check in, Tyra, Mercedes. Are we ready to go?
Yes.
Thank you, and good morning, colleagues, and welcome to Module 2 of our signal of our African Union Smart Safety Surveillance Capacity Strengthening Program. This is now Module 2 on signal detection, and we'll be joined by Claire Davis, who's a leading scientific assessor at the MHRA and has been working on the 3S program for the MHRA for the past few years. Just struggling to move the slides on. Have I got control? Okay. So as before, we are joined by a number of colleagues across the African Union. Sorry, can I just check, Mercedes, am I in control of these slides, Tyra? They don't seem to be moving for me. That's not me, is it? Sorry, bear with us, colleagues. We're having a bit of a technical hiccup with the slideshow, which was fully tested before we joined.
Have they disappeared?
They disappeared from my screen.
Okay, let me find them again. Have they appeared again?
I can see them. They're not on presenter mode.
Okay. Can't even get them back in. Yeah, I keep going back to this mode. 1 second. Oh gosh, can't drop my screen. I can't get the panel off.
All right, if you just go back to the mode that you were in, then we can swap kind of presenter modes there.
Yeah. 1 second while I get this up.
Bear with us, colleagues. Sorry about this.
My slideshow's just crashed on me.
Okay, I have the slides. Let's see if I can share my screen.
I'm going to try again.
I don't know, Mercedes, do you have a copy of these slides you could share? If Tyra's...
I'm just trying to open them now. Yes.
Again, apologies for those who've joined us and are waiting. We will resolve this very quickly. Bear with us.
I might have it up now. Is that working now?
Well, this is a PDF, so it's not great, rather than a PowerPoint. Okay, that seems to be working. Is this Mercedes in control?
Yes.
Okay, maybe Mercedes, if you don't mind, we could just say next slide rather than maybe interfere with the technology.
Sure.
So if you can go to next slide. Please. So if we just pause there, thank you. For colleagues who weren't on module 1, you will maybe benefit from a quick overview of why the MHRA are involved in this capacity building. So just briefly, MHRA are the Medicines and Medical Devices Regulator for the United Kingdom. We are part of the Department of Health and Social Care. We have around 1,200, just over 1,200 staff at the MHRA, primarily based in Canary Wharf in London, although we haven't been there for exactly 11 months due to COVID restrictions, but we do have offices in London. We also have the National Institute for Biological Standards and Control in just outside of London in Hertfordshire. This is an important part of the MHRA's organisation, particularly now with COVID vaccines, as they are the standards body who approve the release of the batches of the vaccines. So they're very much in our thoughts at the minute. Next slide, please. So at the MHRA, our vision is that we promote and improve the health of millions of people every day through the effective regulation of medicines and medical devices. When we say millions of people every day, we are primarily concerned with UK citizens, but we also have in mind that vigilance, the safety of medicines, the understanding of the effectiveness of medicines, and supply chain issues are global considerations. So whilst our primary objective is the health and improvement of health of UK citizens, it's very important that we work globally to share information with colleagues, to pick up trends and issues that may be identified not first in the UK, but will benefit UK patients And similarly, if we pick up issues in the UK, we share those with international colleagues because it will also be concerned in other countries. Next slide, please. So we joined the 3S program a number of years ago. 3S has been working across the globe for probably, I think it's in its 4th year now. And we joined it because we truly believe that pharmacovigilance, if done well and is planned proactively, it will enable the introduction of innovative treatments and vaccines to enable us to tackle public health challenges, such as we are seeing now with the COVID situation. The ability to introduce COVID vaccines and treatments for COVID are supported by risk management plans and active pharmacovigilance systems that give us confidence that we can put these medicines into the public domain, medicines and vaccines, earlier than may otherwise have been the case. Sustainable pharmacovigilance systems should be integrated locally to enable decisions on benefit risk to be relevant to local healthcare settings. And we've been sharing some of our experience on benefit risk with colleagues over the years. A good pharmacovigilance system maintains public confidence. And if you just go back to that previous slide, please, Mercedes, this last bullet point, understanding how the system works gives public confidence in medicines and vaccines. Never more important than at the minute with COVID vaccines. We're addressing vaccine hesitancy, some misinformation. A good pharmacovigilance system and the transparency over the data in that system and explaining what it means gives public confidence in medicines and particularly in our thoughts at the minute, vaccines. Next slide, please, Claire, Mercedes. So as I say, MHRA have been working on 3S. We were invited to join the 3S program because of our background in that we've been a long-standing member of the World Health Organization's International Pharmacovigilance Drug Monitoring Program. We've led similar projects in Europe. If you're interested, you can look up the SCOPE EU project and WebRadar, and you'll find a lot of information around pharmacovigilance capacity building. We work with ICMRA, which is the International Coalition of Medicines Regulatory Authorities, particularly on pharmacovigilance. And we've done a lot of training via 3S and other programs with other regulators globally. So we're very much keen to further this experience, work with the Smart Safety Surveillance Program, the African Union Development Agency, to really see how we can help strengthen pharmacovigilance in the African region. Next slide, please. This is, I won't labor on this. This is some of the reasons that we brought in is as a technical partner, advising the African Union Development Agency and member states on end-to-end pharmacovigilance systems from data management, signal detection, risk management, risk assessment, and all regulatory aspects, including guideline development. Next slide, please. I think the biggest success criteria is working well with colleagues and collaborating in the spirit of teamwork. And we've really seen this already in the AU3S program. And I would encourage colleagues on this call to collaborate, use the chat box, use the Q&A facility to ask questions. I think there's a slide on the functionality of this Zoom meeting in maybe the next slide, if we can move forward. So where we are now, we've had the first module. First module was around data collection. Module 2, we're now moving into on signal management and signal detection, starting today with causality assessment, moving in next week to issues around signal detection and signal management, and we'll be recording a signal management meeting for sharing. Then in March, we'll move into benefit risk assessment, then into April with safety communications, and also be looking at how an expert advisory committee is run. Next slide, please. So this is more of a breakdown of module 2. Today, as mentioned, causality assessment. Next week, signal detection, specific considerations in signal detection. And then on the 24th, an overview of how a signal management meeting is conducted using the UK as an example. So our aims are to provide that high-level overview of how causality assessment is carried out, understand signal detections and the key principles of signal management. Next slide, please. So using the Zoom functionality, if we can use the Q&A button to submit questions to either myself or Claire throughout the webinar, there will be a special holding spot for Q&As. The chat box should be used. We see a lot of colleagues using it to say hello to friends, but if we can use that not to ask questions of the presenters, but to inform the 3S program of any technical issues. You'll all be on mute. There isn't a facility to unmute you, so if you have questions, please do use the Q&A box. Next slide, please. So if we now hand over, thank you, Claire Davis, who's Senior Scientific Assessor in our Benefit Risk Management Group, to take us through causality. Thank you, Claire.
Hi, good morning, everybody. Thank you, Mick. Can you hear me okay?
Yep, very good, thank you.
Thanks. So as Mick says, I'm Claire. I'm a Senior Benefit Risk Assessor in the Pharmacovigilance Department at MHRA, the UK regulatory agency. I have more than 20 years now of working in pharmacovigilance at the UK. So I hope that I have some useful information to share with you today. So this morning, I'm going to give you a general overview of causality assessment in pharmacovigilance. Causality assessment is fundamental. It's crucial to any pharmacovigilance assessor and team, and so basically this session is to provide you with the basic fundamental kind of concepts of causality assessment, because you'll start to see the application of this in subsequent sessions and modules as we go through the training. So the causality assessment introduction that I'll give today will include an introduction to the Bradford Hill criteria. It'll consider other things that we need to think about alongside Bradford Hill, including confounders and sources of bias. And then we'll think about what the key data are in terms of what's needed to perform a robust causality assessment. And then what happens when we don't have robust data, and how do we conduct the assessment. So we'll talk a little bit about methods for causality assessment, use of individual case safety reports. Next slide, please. So if you have a look at this cartoon, the little boy there, this is a picture of a family on an aeroplane, and the little boy is saying to his mum, "I wish they didn't turn on the seatbelt sign so much. Every time they do, it gets bumpy." So the child has obviously mistakenly attributed the turbulence to the seatbelt sign, which, whilst associated, the seatbelt sign is not causing the turbulence. Now, this is essential in terms as a demonstration that association is not always causality. And the little boy's mistake is obvious to anyone who has experienced turbulence whilst on a flight. But how do we determine what is the causative factor and what's the effect when the situation is less obvious? So establishing a causal establishing whether an observed association is a causal association is a crucial concept in public health in general and epidemiology. And if this is because if we can attribute a risk factor to a disease effect, we can prevent or minimize that effect. So if we extend that concept to pharmacovigilance, if we can establish if an adverse event is in fact an adverse drug reaction or an adverse reaction to a vaccine, where a causal relationship is at least suspected and is not a coincidental event, we can take regulatory decisions and appropriate action to minimize risk. Next slide, please. So, causality assessment in pharmacovigilance is essentially the evaluation of the likelihood that a particular drug or vaccine is the cause of an adverse effect or adverse event, apologies. So it's a crucial step in any signal or risk assessment, and as such is key in supporting regulatory decision making. If you don't know whether there is a causal relationship between a drug and an adverse event, how do you decide whether the drug poses a risk to public health and justify taking regulatory action or not if you decide not to take regulatory action? So it can be performed on single cases where it is just that, assessing the causality of a single patient's experience of a drug and an adverse event. It can be done on case series or case clusters, where essentially you're looking for consistency and patterns in the information provided in the reports. And these patterns or information could be things like consistent time to onset, consistent outcomes, or even particular subgroups of particular risks, so maybe elderly or particular disease groups. It can also be done in relation to epidemiological data. When you're reviewing the results of an observational study, be aware though that a statistical association does not necessarily mean that there is a causal association between the drug and the adverse event of interest. Because in this situation, you must also assess the quality of the study in terms of the study design, the methodology, including inclusion-exclusion criteria and statistical analysis methods. Have the confounders and sources of bias been accounted for in the study design? And it's only really after you've considered that can you really have a view on whether the statistical association could be indicative of a causal association. So as mentioned, causality assessment is critical in signal detection and risk assessment. In individual cases, causality assessment can also be used to identify missing information. For example, when I'm reviewing a submitted case for the first time, the adverse event reported may not be a recognised side effect of the drug, but at some point in the future, you might need to perform a signal or risk assessment on a case series, reporting this drug event combination, in which case I ask myself if I need to review the issue in the future, what information in this single case is missing? What information do I need to make that causal assessment and what's missing? And that then informs whether I need to follow up the case with the reporter and ask for that information that's missing. So causality assessment's not always easy. Adverse events may have many possible causes and even contributory causes as well. So a drug might not be the only cause, but in combination with a specific disease or interaction with another drug or a genetic polymorphism, that is when it becomes a cause of something, but in combination with something else. Next slide, please. So as it's complex, people have come up with a number of tools to try and standardize and make a systematic approach to causality assessment. And probably one of the most famous of these is the Bradford Hill criteria. So this is a tool we use every day at MHRA to assess causality. These are widely used in pharmacovigilance generally and also for public health research as well. So the criteria are a group of 9 principles, and these were established in 1965 by Sir Austin Bradford Hill, who was an English statistician and epidemiologist. So these are essentially a useful framework against which to assess whether an observed association between an exposure and an outcome is likely to be causal. So the 9 principles, as on the slide, are the strength of the association, the consistency of the findings, specificity of the observations, the temporal relationship, time to onsets, etc., the biological gradient or the dose response. Is there biological plausibility? What about coherence? And can you test your causation, your association? And analogy. So I'll just go through now these principles in some more detail. Next slide, please. So just a few bits of information, bits of pointers about the Bradford Hill criteria. They can be applied to individual cases, case series and epidemiological studies. So they're quite a good skill to have, a good knowledge to have. The systematic consideration of each criterion helps to build evidence for causal relationship, but it's important to know that though you don't have to fulfill all of the 9 principles in order to establish a causal relationship. But in general, it's accepted that the more you fill, the more likely it is that there's a causal relationship. And as I mentioned previously with regards to epidemiological studies, these are guiding principles only. There are other things that need to be taken into consideration after you've considered the Bradford Hill criteria before a final thought on causality can be made. Next slide, please. So the first of these is the strength of the association, and sometimes this is referred to the effect size. And in general, the stronger the association, the more likely that there is a causal relationship. And this criteria is easier to apply to statistical data where the strength of the association is calculated, so pharmacoepidemiology, where you have things such as the risk ratio, incidence rates, and odds ratios. So the larger the association between exposure and outcome, the more likely it is that the effect is causal. So by this means that the less likely the association is due to other factors or chance. But how, as mentioned before, the quality of the study must also be considered. It's also important to remember that a small association does not mean that there is not a causal effect. And so just as an example of this, I've put a little table up here, which is of the group of statin drugs which are used to treat hyperlipidemia. You may recall that cerebrastatin, the drug that's highlighted at the bottom, this drug was removed from the market in the early 2000s because of a risk of rhabdomyolysis and renal failure, leading to deaths. And you can see here that the strength of the effect size for cerebrastatin is greater than for the rest of the group. Now, all of these products, all of these drugs are known to cause rhabdomyolysis, but the causal association is much more obvious for cerebrastatin than it is for the other drugs in the class. Next slide, please. So just continuing on the strength of the association, for spontaneous adverse drug reaction reports, we can use things like the number of cases in the database or disproportionality analyses to look at effect size. And some of these that we've used in the past and continue to use is the proportional reporting ratio, or the PRR, the empirical Bayes geometric mean, which is actually what we're now using more routinely in our standard signal detections. And this tends to be more stable at lower numbers of reports compared to the proportional reporting ratio. And we also now, as standard, and particularly for vaccines, which and also at the moment for the COVID vaccines, we are routinely doing observed versus expected analyses on our spontaneous ADR reports. Next slide, please. So the next Bradford Hill criteria is consistency, also referred to as reproducibility. And this refers to repeated observations of an association in different populations and different circumstances. And this strengthens the likelihood of a causal effect. So, for example, some of these might be that you see the same association in different studies of different designs, studies performed in different locations, or in different types of studies from non-clinical, clinical and clinical data, for example. Maybe you see the same association in different countries and populations, or the ADRs are being submitted by a range of reporters, so different people in the community are seeing the same thing. And also there's a consistency sometimes in the pattern of presentation of the observed effect. And I'll come on to that now in the example. But it's important to note again with this criteria that the lack of consistency does not necessarily exclude a causal association, because some effects are produced only in certain circumstances. And again, this would refer to things like drug interactions, or maybe even the effect might only be seen in people with renal failure and not in everybody, for example. Next slide, please. So this is an example of the consistency criteria, and I'm sure everybody will be aware of the thalidomide tragedy that happened in the 1960s and 70s, where women were taken thalidomide for nausea in pregnancy during the 1950s and 60s, and doctors at that time started noticing patterns of babies delivered from women who had received thalidomide in pregnancy, and they observed striking patterns of malformations which had not been seen in combination. So the consistency and the pattern of these malformations had not been seen in the background population prior to this drug coming onto the market. And the defects were primarily of the upper limbs, but also the lower limbs, and the face, eyes, ears, genitalia, internal organs, including the heart, the kidneys, and the GI tract. And the pattern of malformations were observed in different countries as well, in the UK, the US, and Germany, where this drug was being used. So as a result, approximately 10,000 children were born with phocomelia. This was the name given to some of these symptoms because of the thalidomide. And this really galvanized the need for developing national regulatory agencies for medicines. And this is in fact the basis on which the UK Yellow Card Scheme was established in 1964. So next slide, please. So the next criteria is specificity. This is a more specific an association between the drug and the observed effect, the greater the probability of a causal relationship. Now, this historically has been quite a difficult criteria to fulfill when you're doing causality assessment, because there are very few effects which are specific to an exposure in practice. But this is just one of the examples of those very rare specific effects. This is something called nephrogenic systemic fibrosis, which is a rare disease with skin and other organ involvement of fibrosis, a fibrotic reaction. But this is almost exclusively seen in patients with severe renal injury who have been exposed to gadolinium containing contrast agents for MRI scanning. So it is specific to gadolinium use, and it's specific to gadolinium use in patients with severe renal failure. So this is a really good example of a specific causal effect. And some of the other true examples, I mean, it's not necessarily a drug relationship, but one of those would be, for example, asbestos and asbestosis. Asbestos is a substance that was used in kind of building materials and has been found in walls and so on in many countries sort of 10s, 20s years ago. And people who have been working or have lived in close proximity to asbestos have developed a fibrosis condition of the lungs called asbestosis. Now, when Bradford Hill came up with the specificity, it was more difficult to find examples of and fulfill this criteria, purely because at that time things like the investigations, medical science and experimental science and that kind of thing has moved on quite quickly since then. And so there's some more and more ways that you can actually show specificity, which previously couldn't be done. And the asbestos example is just one of those where you can isolate asbestos fibres in the lung tissue of patients exposed. So that's a really good indicator that this is a specific effect of that substance with a specific lung etiology. So, however, there's also very few things have only one cause. And the other thing that you can use under specificity with relation to medicines is people can now demonstrate a molecular mechanisms of actions which previously couldn't really be demonstrated. And so if a drug has a specific mechanism of action and the event also has a very specific pathology, then you can use specificity for causal association in that context also. Next slide, please. So this is the only mandatory Bradford Hill criteria. The adverse event has to occur after the exposure to the drug and not the other way around. If it occurs the other way around, there is not a causal association. And this can just be not just in terms of did it happen before or after exposure to the drug, but it can also be more subtle temporal relationships. And the question you need to ask yourself is the reported time to onset consistent with what is known about the pharmacology of the drug? And not only just the pharmacology of the drug, but also the pathology of the adverse event. So I've put some examples here of this. So you have a report of anaphylaxis 21 days after vaccination. Knowing what you know about anaphylaxis and how vaccines work, is this likely to be a causal relationship? And the answer to that is that, could you click the slides please to reveal the answers? Thank you. So the answer to the first one is inconsistent with what we know about the pathology and of anaphylaxis following vaccination. So in this case, it's unlikely to be causal. And the next one, please. So for breast cancer, 2 weeks after taking a short course of an antibiotic, is inconsistent with what we know about the development and the pathology of cancer, and also about the half-lives of antibiotics. So this again is unlikely to be causal. And for fever, headache, muscle and joint pains developing 24 hours after vaccination, we know that these are typical symptoms of the priming of the immune response after vaccination. And so this is consistent with what we know and what we expect to see. So this is more likely to be a causal association. Next, please. So the next one is biological gradient, also known as the dose-response relationship. And in general, the greater effect bigger effect is seen with increased exposure to the drug, indicates more likely causality here. And some clear examples of these would be morphine and respiratory depression. The more morphine you give, the more respiratory depression the patient experiences. And similar for systemic effects of inhaled corticosteroids. But it's important to remember that this isn't always the case. There are some effects, there is a threshold rather than a dose response, and maybe anaphylaxis would be another good example of this, where exposure to the drug or the vaccine is enough regardless of the dose. Next slide, please. So just going through quickly the remaining Bradford Hill criteria, and the first one is plausibility. You click the slides, please. Thank you. Now, the plausibility is basically the existence of a reasonable pharmacological or biological mechanism for cause and effect. A plausible mechanism, though, is helpful, but at the time that Hill wrote these criteria, he realised that the mechanism should not rule out a causal relationship, because we only know what we know now, and we don't know what's going to be discovered or what will be a new effect with a new mechanism. So it's very dependent on existing knowledge. So lack of knowledge of a mechanism should not rule out a causal relationship, especially if there are other criteria that are met. Events with a higher plausibility of drug causality are things like erythema multiforme, rhabdomyolysis, agranulocytosis, renal failure. So these kind of things are kind of plausible. They're known to be in general, drug related for quite a lot of drugs. So the next criteria is coherence. So this basically means consistency across all data sources. So is your adverse event report, your single case, or your epidemiological study, the results of that, are they consistent with other studies? Are they consistent with the animal data? And so on. And so when you start getting coherence and other sources of data which agree, providing that they are also robust sources of data and don't have other flaws as we discussed previously, then that's a good indicator of a causal association. The next one is reversibility. Some people call this experiment, some people call it reversibility. And some of these things that you can look at to kind of add to your argument for a causal association are things like antibody titers, metabolites, preclinical evidence again, but also dose reduction. What happens when you reduce the dose? Is there a reduction in the event? And again, the more extreme of that, de-challenge and re-challenge data. As we know, if you stop the drug, does the event reduce or diminish or go away? That's not always necessary though. Some effects are irreversible. But the one thing that is quite a good indicator of causality, of course, is re-challenge. What happens then if you give a drug to a patient, they experience an effect, you take the drug away, they recover? What happens if you give that to the patient again? And if they experience the same effect on the second challenge, then that is a good indicator again of a causal association. So the last of the Bradford Hill criteria is analogy. So again, this is this basically in pharmacovigilance context is, what about other drugs in the class? Do existing products that work with similar mechanisms also are they known to cause this adverse event? And again, this is a little bit like plausibility, where it is slightly limited by current knowledge. So I think the main message to get across with Bradford Hill and any of the tools that I'll come to discuss is that these are guidelines only, and they still require judgment of the assessor. And so they're quite complex considerations. Next slide, please. So causality assessment would really not be complete without consideration of confounders, which are alternative explanations for an observed association. So confounders are essentially things like underlying disease, medical history, concomitant medications that could cause the effect, and also socioeconomic things like smoking, alcohol, body mass index, and occupations, and so on. Bias is another thing that you need to consider in terms of epidemiological studies in particular, but also in terms of ADR reporting rates. We know that in the UK that publicity around a certain adverse event, for example, can influence reporting rates for a particular drug. Disproportionality analysis have these been done with the appropriate background comparator? So if you have a database that is largely vaccines and you're looking at a possible causal effect with a drug, do drugs and vaccines generally have the similar side effect profiles? And is your vaccine data set the right comparator when you're looking at a drug effect? And similarly with observed versus expected, which I mentioned we're doing a lot of at the moment with the COVID vaccines. If you're looking at all-cause mortality within the background population, are you comparing what you're seeing with your vaccine to the right background population, or are you comparing it to the whole country population, or should you be comparing it to those who are receiving the vaccine? So is it a childhood vaccine, in which case you would look at all-cause mortality in your background population in children, or is it primarily an elderly, frail population that you're vaccinating, in which case you would need to do all-cause mortality calculations in the appropriate background population for comparisons. Next slide, please. So this is just a demonstration of a confounder. These are variables which are independently associated with both the exposure and the outcome, and one is they're not on the causal pathway from exposure to outcome. So this example is of a statin, which these are used to treat hyperlipidemia and prevent cardiovascular adverse events associated with that. And in this case, the confounding, the outcome of interest is myocardial infarction. And the confounding variable in this case is the underlying illness itself, in that hyperlipidemia itself is a risk factor for myocardial infarction. So in this case, hyperlipidemia is the confounder between the signal between statins and adverse event of myocardial infarction. Next slide, please. So moving on to the key data for causality assessment. I mean, it's important to include as much information as possible in an adverse event report so that causality assessment can be performed. And as assessors, what we really need to do as well is request follow-up data if needed. And we can also support with external data if we can't get the information from reporters. So things like published literature, background incidence rates of events, and occurrences from other databases and registries, for example. So in an ideal world, every ADR report would be complete. And this is some of the information that is important for causality assessment. And that includes medical history and concomitant illness, which are, often common confounders. Other risk factors, which depending on the event, include alcohol intake, smoking gain, occupation, rural or urban living, other drugs, the start and stop dates of drugs. That would help you determine temporality and whether it could be another drug that was started just before the onset of the adverse event, and rather than the drug that's been reported. The indication for use, often as we've just seen with the statin and hyperlipidemia example, is a confounding factor. And then, of course, what's also important is the description of the event, the clinical description and the results of investigations and performed. These are really important for, all reports really, but for vaccine type reactions as well, because, for example, some of the adverse events of interest with vaccines are often require quite complex diagnoses for things like transverse myelitis, or some of the other neurological type effects, or Bell's palsy, or Guillain-Barré syndrome. These are quite complex diagnoses in clinical practice. Can you follow up and ensure that actually this is what the patient experienced, or did they experience something similar? So the other just information, evolutionary of event, including dates of onsets and the end dates, severity and seriousness, outcome, and re-challenge information as well is really useful if re-challenge is appropriate and was performed. Obviously you would never re-challenge where it's not safe to do so or ethical to do so. So how do we take all of this information and kind of come up with an answer for a causal association, particularly if you've got missing information and you can't fulfill the criteria? So there are further tools for this. So next slide, please. So these are methods for individual case reports. And they're basically split into three kind of broad categories of methods. There's the first one, which is expert opinion, which is based on the judgment of individual experts or assessors. It's quite subjective, and as you can imagine, is variable and not really reproducible, or can often be inconsistent depending on which assessor is doing the assessment. So then we have things like algorithms, which follow decision trees, but is also defined and informed by expert and pharmacological knowledge. It's more standardized than expert opinion, so is better at kind of coming up with more reproducible events or outcomes, but it's still subjective because of the input by individual experts, and the reproducibility here is low. So probability assessment is probably the best of these, but it's incredibly complex and usually requires kind of computer software, numeric data in order to do that. Next slide, please. So there are many algorithmic and methods that are available, and these, I've just put this slide together for your future kind of reference, and if you're interested you can look these up. So it just basically demonstrates how many different methods there are of doing causality assessment on individual cases. All of these use some elements from the Bradford Hill criteria, but they have emphasis on different aspects of the Bradford Hill criteria. Some of them, for example, the WHO-UMC system and others, they also take in consideration the quality of the data and the completeness of the reports as well. So for those who are interested, the reference at the bottom of that slide provides a summary of the strengths and the weaknesses of each of these methods. Next slide, please. So this is just a very quick overview of the WHO-UMC system. Some of you may very well be using this already. This is basically takes the reports, and then based on the answers to these questions, you get 6 causality categories of certain, probable, possible, unlikely, conditional, or unassessable. And this list is of assessment criteria with the emphasis on, with the Bradford Hill criteria of time to onset, the role of other drugs and diseases, temporality, and de-challenge, re-challenge. But it also takes in consideration case completeness and case coherence as well. Next slide, please. So this is an example of an algorithmic scale. This is the Naranjo scale for individual case causality assessment. And this is based on a summary score. A summary score is based on the answers to 10 questions. So if you have your report in front of you and you ask yourself these questions in turn, and depending on the answer, you attribute a score to that answer, and at the end, add up the sum of the answers to the 10 questions, and you get 4 categories of certain, probable, possible, and unlikely. So the limitation of the Naranjo scale, though, is that it doesn't address main points that are necessary for causality evaluation of drug interactions. So essentially, there are the kind of message that I want to give from this is that there are lots of tools available. They all have their own strengths strengths and weaknesses depending on what it is you're looking at. Some are more appropriate for certain causality assessments. There's the Rackham scale, for example, which is designed to be used for specific outcomes of interest, things like skin reactions and liver reactions, for example. And there are many like those. The difficulty has been that there has never been one that has been universally accepted as the scale to use because of these differences and the reproducibility and the inconsistencies in some of the answers that they give. So overall, I think the message is that these tools are there to help you and provide a framework for thinking through the structured thoughts that need to be considered when you're doing a causality assessment, but they should not be used in isolation. You still need to use quite a bit of expert judgment, your own judgment, to come up with a causality assessment. So next slide, please. So just to sum up, challenging, very challenging to do causality assessment. It's not an easy task, but I find it quite a rewarding element of pharmacovigilance. It's kind of a bit like the picture detective analogy, or similar to kind of making a diagnosis in a clinical setting. You take the information you have, you think of what other information you need, and then you go out and find that before you come up with your conclusion. So generally in pharmacovigilance, particularly for spontaneous reporting systems, we will never have all of the information that we need for a perfect causality assessment. So you just have to do the best that you can with the information that's available to you, and use on your own experience, your own judgment, to make an informed decision. So your assessment and decision, though, should be defensible, backed by evidence. And so I always think about this, if somebody were to challenge my causality assessment, my decision to take regulatory action based on my causality assessment, could I provide the evidence retrospectively to convince that other person that my judgment on causality is sound? And as usual, the tools are great. They're there to help you think logically and consistently through those judgments. But as I mentioned before, there are other things to consider, and beware of confounders and bias, which can also interact with your causality assessment. So thank you very much for your time and for listening. I think that's the end there for me. Thank you.
Thanks very much, Claire. It's certainly not the end, because there's a number of questions that have come in. I might start with one of my own though, because we know that many of the countries that we're working with in AU have fairly low reporting rates. So when you've only got 1-2 cases that are maybe quite strong on their own, how do you go about strengthening that evidence when the volume of cases is low?
I think it would really depend on what events and what exposure you're looking at, because of course, because that will always also determine what other sources of information are available to you. So for example, if I had to do ICSRs, I might then look to the literature. I would probably look to looking at other drugs in the class. So basically go out looking for other information that could be supportive of a causal association, mechanistic wise as well, that kind of thing. Is this plausible? So it really does come back actually to the Bradford Hill criteria, even when you've only got 1 or 2 cases. And I think in terms of making regulatory decisions based on 1 or 2 cases, that again comes down to the events in terms of how many people are taking the drug, what is the overall public health risk in terms of size, the seriousness of the risk, and that kind of thing. So, with the vaccines at the moment, millions of patients, of recipients, of I think 15 or 16 million people now have received the COVID vaccine in the UK. So 2 or 3 reports of muscle ache is nothing, and kind of instinctively you know that it's probably related, it's listed. But if you've got something like immune thrombocytopenia, for example, which can be typically vaccine related, just 2 or 3 of those cases, even in a background of 16 million vaccinations, would make you sit up and look further at those and look for other information and other ways to verify that from observed versus expected analyses, for example.
Thank you. I think we'd also encourage regulators to look in other regulatory databases or ask other regulators. And of course, there's the WHO Vigibase as well that's run by UMC. So I think that the message is you're not on your own. You may have 1 or 2 cases in country, but talk to other regulators, look at other data sources to see what others have had reported to them as well. Some number of questions coming through the Q&A box. If we'll just work through them in turn. Some of these will also be picked up in the next couple of sessions around signal detection and signal management. But the first one here is, could you please explain how to perform the EBGM in determining the strength of association? I know that we'll be covering these kind of statistical methodologies in the next session, but do you want to touch on that, Claire?
I think basically the answer to that question is we don't. The computer software in MHRA does that for us. And you'll see a little bit more of that from Lily in the next session on our signal detection methodologies, is that actually it's not something that we actively do as assessors. It's run through our signal detection software. So it's quite a complex statistical calculation. So yeah, we wouldn't be able to do it for every single event combination that are reported.
Yeah, but the EBGM will be part of our data mining run, and it's really a flag of disproportionality to say to the assessor, this looks disproportionate when compared to the rest of the database, please investigate. So we will cover that in more detail in the next session.
So just to add to that, Mick, though, for example, our threshold for a signal, if you like, for looking into further is a EBGM of 2.5, and then the 95% lower confidence interval of 1.8 when you've got 3 or more cases. So we have thresholds for those statistical values in terms of what we would then look at in more detail.
I see a hand raised. Unfortunately, we can't go to raised hands to unmute you, but if you've got your hand raised because you've got a question, please put that question in the Q&A box and we can pick that up. The next question, Claire, is do causality assessments, are they conducted by a broad team of experts? Are they done individually or by a team, and how does it work at the national level?
So essentially, I think some of this will be covered a little bit in Lily's session again, but essentially it initially is done by the individual person, but there's always a level of discussion around that within a team as well. So an individual assessor will make that initial causality assessment and present it in an assessment report to a wider team of medics, pharmacists, epidemiologists, who then agree or disagree with that assessment. And often, depending on what the issue is as well, we will present the assessment to experts, external experts of in their field, and that will come back to our further sessions on seeking expert advice as well. So essentially, initially, it's a kind of a phased process depending exactly on what it is, but essentially it's one person to start with, followed by a team.
Just before moving on, I see in the chat box there has been requests for the slides, the materials, both from this session already and previously in module 1. These are available, and if you haven't got them already, we'll be in touch to share or point you towards where you can access them. Claire, next question is, among the different methods for conducting causality assessments, which ones have a strong association and causalities to be used for decision making? For example, the person asking the question here says they use the WHO algorithm.
I mean, I think probably the WHO algorithm is used by many people. It's standardized and it's widely accepted. So I think it's a reasonable one to choose. But as with all of these tools, you always have to bear in mind the weaknesses of them and where you need to kind of do additional kind of research, if you like. Do you need to find additional information and that kind of thing? So I think as long as you're using the tools in the knowledge that they're not that you need to take into consideration other things as well, that they're not perfect, that they do have limitations, then I think yeah, the WHO one is a really good place to start. But just to say as well that at MHRA, because of the volume of cases that we have, we don't really do individual case assessments unless we've detected a signal, and then at that point we might look at individual cases. But we don't generally use a structured tool for that. We seem to have the kind of the Bradford Hill criteria plus confounders and bias rather than a tick box kind of algorithm type thing.
I think you've touched on the next one already, but it's around the use of literature reports. Is it okay to check for experiment details in literature reports?
I think so. I mean, again, you have to take the that literature report. You'll need to have a look at how robust that experimental data is again. So, depending on what experimental data it is, you might have a non-clinical assessor look at it. For example, if it's animal data, because of course all studies will come with their strengths and weaknesses as well. So I think it's fine to go there looking for this kind of thing. But as with everything, there needs to be an assessment of the robustness of that data you're seeking and using in your arguments.
And then we have, what are the ways to avoid or control confounding effects when conducting causality assessments?
I don't think you can when you're looking at individual, spontaneous reporting data. The only way you can kind of control for those is if you're designing a study, and then you would do so in your methodologies and your statistical kind of analysis, whether you adjust for confounders and channeling and bias effects, for example. But I don't think you can, because spontaneous reporting is all about it's non-interventional, so we don't control for it. But usually if it's clear that there's confounders in them, then we wouldn't consider it a strong case, and it wouldn't be added to the evidence for a causal association. For example, we might exclude that from our further analysis if there were clear confounders in a case.
I think you've actually touched on something that will come up in future sessions as well, and it's around and we covered some of this in module 1 around the limitations of spontaneous reporting and the need for other tools in the armory rather than just a reliance on spontaneous reports. Do you want to say more about that?
Yeah, and I think so. It's becoming ever so more clear actually with the vaccine work that we're doing at the moment. I'm currently part of the Pfizer COVID vaccine safety team. And although we're looking at all of the yellow card reports that comes in, they're very, very, very difficult to do causality assessment on individual cases for vaccine reports, because you can't do you can't re-challenge a vaccine. There's no kind of dose effect. So a lot of the Bradford Hill criteria don't actually apply to vaccines. So much of what we're doing now it's about detecting kind of disproportionality with with spontaneous reporting. But then we're looking at other ways. If we find a signal, if we find something that's looking a bit, oh, we need to have a look at that, we then go into other databases. We ask other regulatory authorities whether they've seen cases. So yeah, again, no, it's not a reliance. It's there as a useful early warning tool. But we go to other sources of information. So, for example, we're using CPRD data, our clinical practice research database, for background rates and things like that. And quite quickly, we can perform studies using that data with the new vaccine data that's coming in as well. So it's all about comes back to the Bradford Hill criteria of coherence, where are you seeing this across different sources of data, for example? So, yeah.
Interesting one next around genetics. Can genetics be part of confounding factors? Before you come in, Claire, it might just be worth sharing some of the work we're just getting started at the MHRA, which we are calling our Yellow Card Biobank. The plans that we have is that when we get yellow cards for particular drugs or adverse events of interest, that we will be collecting genetic samples and building up a database of these samples, so that we can understand better the role that genetics might play in certain patients' predisposition to having certain adverse events. So it's certainly something that we think needs to be investigated more, and we'll be moving forward over the next couple of years to develop this biobank, so we can be better informed around the role genetics play. I don't know, Claire, if you've seen anything already with regard to genetic factors.
Nothing that springs to mind in terms of a tangible example of genetic factors in terms of being confounding factors. I mean, obviously it can be if it predisposes you to a particular outcome, for example, genetics, then that actually I do have an example. For example, people who have congenital long QT syndrome are at higher risk of sudden death from cardiac arrhythmias, for example. So if you then have a patient or a report of somebody who's died very suddenly with cardiac arrest with no other kind of comorbidities, then they have a family history, for example. This is actually a report that came in yesterday of their father having died from a sudden cardiac death early in his life as well. So that would be a genetic predisposition, and actually the advice for that is that they would be undergoing kind of family screening for congenital long QT syndrome. So that's just one example of that. But I would say, yes, it can be part of confounding factors purely because it would predispose you to certain adverse outcomes if that's the case. But also as well, it can be genetics can be equally contributing factors with drugs. I mean, polymorphisms in drug metabolism, for example, is a prime example of where drugs interact with genetics to cause adverse events or increase toxicity or even reduced efficacy in case of codeine, for example, and prodrugs.
Thanks, Claire. The next question is around the AEFI reporting form, and the question is, how important is it to have an alternative diagnosis section in the reporting form? I don't know if there's
I'm not sure necessarily. It depends, I would guess, on the confidence of your assessment team. I think it would definitely be helpful information to know that your reporter has thought about other causes, but there are other ways that you can kind of collect that in the forms anyway, and without having the reporter themselves suggest what the alternative diagnoses are. If you have the information in other parts of the form, you can often do that yourself. So it depends really on how thorough reporters are generally, and maybe it just makes people think harder about reporting as well. So but it's not something that we necessarily do in the UK in the spontaneous reporting cards. So we don't really have experience of using that ourselves. It's work we have to do ourselves anyway, if you like, through what's missing and then following up with reporters.
Yeah, I think this may go back to some of the areas covered in module 1 around the importance of the quality and completeness of reports and the need to do good follow-up when quality and completeness is maybe not optimal. So this whole program is building from module 1 around data collection, module 2 around signaling, then assessment, and so on. So I think it might be worth, if you haven't got the materials or weren't present for module 1, to access those resources and have a look at that, because the quality and completeness of a report really does aid our ability to detect signals. So the next one, what is the next level of hierarchy if the causality assessment's confidence level for expert analysis is fairly low?
I'm not sure if I completely understand this question, but I'm interpreting it as being if your causality assessment is that you don't think, or it's unlikely to be a causal association. I don't think there is necessarily a level of hierarchy. I think it's, if you're not seeing a signal, you're not seeing a signal. That would be my conclusion, as if you have a case series and none of the cases in that series are providing good evidence of a causal association because they're not meeting the criteria, or you have a lack of information on which to do that, then you would just keep on monitoring that for the future if you get more cases. So it's just a continuous thing. It's the pharmacovigilance life cycle, where if you don't have enough information at this point in time, next week you might have that information and your assessment will be different. So it's a continuous kind of process. It's not necessarily hierarchy, but again, you can go looking for that information. If you think it's plausible, for example, biologically, pathologically, and pharmacologically, then again, you might want to supplement your review of your individual cases with a literature review, or and so on. So it's about building a case, really, and if the information you've got is weak, then you don't have enough evidence on which to make any kind of regulatory recommendations, other than wait and see for more cases. I think at that point it would be nothing here at the moment, but we continue to monitor.
Yeah, and I think some of this will come up in the next sessions around signal management, in the sense that sometimes you park a signal waiting on further information, more cases to come in, and then you review that signal once it's been triggered again. But as you say, there's always the ability to check the literature, get expert opinion, look at other data sources such as UMC's Vigibase. And also I think the manufacturers have a role to play. So if we've seen a couple of cases, we would often go to the manufacturers, the marketing authorization holders, and say, we've seen a couple of cases here. What are you seeing? Have you seen this either in the clinical trials, any studies that might be ongoing? So there's always that option, and recommended option, to be in continuous dialogue with the marketing authorization holders, because they'll have pharmacovigilance responsibilities also.
I think there is a threshold to be reached, though, because of course you're not doing all of this for every single case reports that comes in the door. So there is kind of a level of confidence, if you like, at which you want to take things, if you want to kind of spend the time and the effort and prioritize how and you're going to take that forward.
And again, I think that issue around prioritisation will come up in the next sessions, because we've got to be realistic. We have limited resources. You need to be identifying what are your priorities and how to set timelines and activities around what's the most important and pressing.
Yeah.
The next question, I'm curious what you mean when you say you don't do individual case analysis. I assume you mean that you have the data collection tools and you do a purely mathematical analysis. I think it's a combination of everything, Claire, isn't it? Where we will look at individual cases for certain drugs, particularly if they're new to the market, or for certain reactions, all fatal cases, all pediatric cases, for example. Do you want to explain more about that? And again,
yes, so this a little bit more about this will be Lily will cover in the signal detection processes that we do. So essentially, we don't do individual case analysis in that the case comes through the door and we perform an assessment, a formal assessment of every case. What happens is that those cases go into our database, and then once a week we run a statistical signal detection software on the new reports in our database. And if they hit a threshold level, we use the EBGM, as I mentioned before. If it's EBGM of 2.5 with a lower confidence interval of 1.8, and then there are more than 3 cases, then we will look at that, and we will look at the individual cases that make up that case series. But we do also have lower thresholds for certain types of reports. So we will look at all fatal cases, and we will look at all new pregnancy cases. We will look at all new cases in children. We will look at all cases where these kind of adverse events of special interest that are generally known to be drug related. So things like acute renal failure, rhabdomyolysis, thrombocytopenia, liver injury, these kind of things that are quite often drug related. We will look at all of those cases every week for every drug in our signal detection. So we don't do an initial review of individual cases. It's once we're sort of reaching our signal threshold. And I mentioned the thresholds are different for different types of drugs and different types of events, as per a standard operating procedure. So we have a framework which everybody kind of works to on that.
The next one is wanting to find out more. You gave an example there about a vaccine not being the cause of breast cancer 2 weeks after administration. Adama is asking to find out more how you established that fact.
I think that is largely based on what we know about the pathology of breast cancer. I think it's very, very unlikely that knowing what we know about the time, the latency onset of cancer, that 2 weeks after having a vaccine, that could be the cause of the breast cancer. It's very likely that that breast cancer had been developing for a lot longer than before the vaccine was having. So again, that's based on what we know about the pathology of breast cancer itself.
Thanks, Claire. I think the final one here is probably for me and others, but outside of the session. What's the plan to strengthen ADE reporting systems, specifically in Africa? So as part of the AU3S program, we are working with the African Union Development Agency and the Bill and Melinda Gates Foundation to support strengthening ADR collection, the provision of new tools such as the Med Safety app, the addition to the Med Safety app of reporting forms for AEFIs, but not just the tools. We're working with the national regulatory agencies around some of the communications that they use to encourage people to use those tools, and then how to manage the data when they receive it. So there's a whole strengthening program, not just on ADR reporting, but the whole pharmacovigilance system. This is in its infancy now, this program, but the Med Safety app as a reporting tool is being rolled out in a number of countries. Specifically at the moment, we're focusing on COVID vaccine reporting in Ghana, Nigeria, South Africa, and Ethiopia. But we do know that the African Union Development Agency have much more ambitious, longer-term plans to extend that reach and extend the technologies that are used to support both ADR collection and the management of those reports once collected. So we have no more questions in the Q&A box, which is because we're right on time. Actually, we've probably just used the last couple of minutes. We'd ask you to complete the polls. We collect after every session some feedback from you through some online polling. So you'll see the questions come up. I think you have to answer all of them before you can click submit. So please take the last couple of minutes to complete these couple of questions. Thanks so much for your attention today. And Claire, thank you very much.
Thank you.
Thank you, Mick and Claire, for providing a fantastic webinar. Just to let everyone know, as with previous sessions, the materials will be circulated, and so will a copy of the recordings. Module 2, session 1, as you know, is completed. Session 2 and session 3 will be taking place next week. We have a further set of modules for module 3 to 5 due to take place later in the year. Dates will be confirmed and invitations will be circulated. We'd like to thank everyone for attending the webinar today and look forward to welcoming you to the future webinars for the African Union Smart Safety Surveillance Programme. We'll leave the poll running for a few minutes, but the webinar is now finished. Thank you very much for attending.